I'm not sure if I can record session. Okay, let's see if we can continue with the lecture. So I'm going to share my screen now. So did anyone know where do we stop last time? Anyone? Nur Fitria, where are you? You know where do we stop last time?
I think until classification of hormone.
Very good. Okay, so we stop up to classification of hormones here. Okay. All right. So the hormones, as we know previously, they are of two main types. Okay. The first thing, or the first type that I've mentioned before, is the steroid base. The second one is the peptide base or polypeptide. Some of you call it protein base. So it's a lipid or the protein base. Mainly these hormones are being categorized based on the structure, okay, based on these two main group. Okay, generally, most of them, okay, and a few of them like from the amino acid or derivative type. So how do we classify the hormones? So we classify them based on the chemistry or the composition. What are the molecules? What are the atoms that build up the structure? We also classify them based on the solubility, how well they're being dissolved in the water. Maybe they are hydrophobic, or maybe they are hydrophilic, okay, in nature. So we need to know which structure, which hormones they refuse to mix with the water and the one that can be mixed with the water. We also classify them based on the location of the receptors. Where is the receptor attached to? Okay, some of the receptors attached to the cells or tissues. Some of them might attach to, maybe not attached, but maybe available inside the circulation. And the nature of the signal used to mediate hormonal action within the cells. Okay, the nature of the signal. It can be, the nature of the signal, it can be direct or indirect signal. Okay, so how do we classify them? Based on these four main features. And in the top left corner, okay, the first row that you can see hormones that bind to intracellular receptors. I give the examples like the androgen, the hormones for the female, for the males. You have the calcitriols, also a steroid base or lipid base hormones. So this is based on the receptors. Okay, so all of these group, androgen, calcitriol, estrogens, glucocorticoids, mineralocorticoids, progestin, retinoic acid, and thyroid hormones, they have to bind to a special receptors on the cells or tissues, right? In order to them to elicit a function or to exhibit the function. The second one is that hormones that bind to the cell surface receptors, okay? So this is on the cells, on the cell surface. The previous one is the intracellular receptors, means the receptor that is located inside the cells, right? So based on the location of the receptors as well, we can differentiate. We can know that they are of different group. Example of the hormones that bind to the cell surface receptor, for example, the second messenger is CAMP, okay? Like alpha-2 adrenergic catecholamines, beta adrenergic catecholamines, ACTH, ADH, antidiuretic hormones, calcitonin, chorionic gonadotropin. Human means HCG, or you can, in some book they put it CG and a small letter H in the front. Okay, call it HCG. This is for the detection of like the pregnancy kit. If you get married and if you got pregnant, so you want to see that in the pregnant lady, the expression of these hormones are very high, especially because they are pregnant. Okay, but if you are not pregnant, then the level of the chorionic gonadotropin hormones is very low. All right. You have another type of hormones. We call it glucagon, lipotropin, luteinizing hormones, melanocyte stimulating hormones, parathyroid hormones, somatostatin, and thyroid stimulating hormones. So these are, you know, the second messenger is the CAMP. When all of these hormones bind to the certain receptors that is located on the cell, for example, beta cells of the pancreas. Okay, when this hormone binds to the cell surface receptors, they will produce the signal in the form of CAMP. Right. So these are the all hormones that can bind to the cell surface receptor, but they can only send signal in the form of CAMP. If, right, if the hormones bind to the certain receptors or cell surface receptor and the signal in the form of cGMP, right? So this is another group of hormone. For example, the hormones are atrial natriuretic factor and nitric oxide. Right, on the right hand side boxes, okay, you can see the C, the second messenger is calcium or phosphatidyl inositol or both. Okay, in certain condition, the signals or the second messenger that is expressed inside the cells can be in the form of calcium. So the calcium is the signals. Some other hormones, they can bind to cell surface receptor, initiate CAMP, or maybe other hormones can initiate cGMP. But you need to know that some other hormones also capable to send signals, okay, in a form of calcium or phosphatidyl inositol. And the final one is that the hormones like adiponectin, FGF, NGF, prolactin, PDGF, these are all the hormones that forms, okay, a signal in the form of kinase or phosphatase cascade. In other words, if you look at all these boxes, you can know that each and every hormones in our body, they have a special or specific receptors that they can bind to. And of course, the signal also are specific. Okay, you cannot say oxytocin like in the box C. All right, oxytocin, they cannot, you know, initiate a signal in the form of kinase. No, it's only capable to initiate a signal in the form of calcium or phosphatidyl inositol. All right, and so on. So again, the main category, regardless what it is, maybe you want to say group 1 is steroids, group 2 is polypeptide. Some books they use group 1 as polypeptide, group 2 is steroids. But you just follow what I'm showing you. So like this in this picture, I'm putting two types. The group 1 is steroids, means these are the steroid based, they are lipid based hormones, and they hate water so much they cannot mix with the water. Whereas in the protein type of protein based hormones, they love the water so much in a way that they can bind and they can be mixed together with the protein. They form a complex with the proteins, and they can travel inside our circulation using these protein-based hormones. In terms of the solubility, of course, you know that this is lipophilic. They love the lipid, okay? They love the lipid condition. And then after that, they can be solubilized, means they can be diluted inside the lipid condition. On the other hand, the protein-based, they are hydrophilic. Okay, they only can be mixed inside the water, and they are lipophobic. Okay, the other way around. Lipophobic means the protein-based hormones, they don't like the lipid condition because they cannot be soluble. It's quite difficult for them to travel. In terms of the transport protein, yes, steroid have this transport protein, but protein-based hormone, they don't have transport proteins. In terms of the plasma half-life, means how long they can survive in the system. It's quite long. It can take hours to few days. Okay, can, you know, up to weeks. But for the protein, you know, the protein structure is very fragile. They are easily degraded due to the pH or due to the temperature. So the half-life is very short. They cannot survive that long. So you need to be mindful of that. In the group 1, regarding the receptors, they can be found in the intracellular condition. Okay, intracellular means in the internal side of the cell. Whereas in the group 2 plasma membrane, okay, that is where, you know, the receptor for the group 2 are most commonly binds. In terms of the mediators, okay, this is the signals in the form of what? In the group 1, like the steroid-based or lipid-based hormones, they form a receptor and hormone complex. Okay, so for the group 2, so they have signals in the form of CMP, CGMP, calcium, kinase, phosphoinositol that I have mentioned earlier, all right? So this box also important for you to know. This is kind of repetition what I've been telling you just now. This is further classification based on the second messenger system. You know, we have the 4 second messenger system. If I ask you inside the examination, tell me which hormones have this second messenger system. So you can see, you can say, or you can answer that it's belong to the lipid or protein-based hormones. Which group? Group 1 or group 2? Anyone? Are you there? Nor is Mawani. What are you doing?
Group 2.
Group?
Group 2.
Very good. Okay, brilliant. So group 2, because it's the protein-based. Lipid-based don't have this second messenger system. What are the second messenger? So the hormones, when they bind to the receptor, they will release the messenger, or they will release the signal in the form of 1, CAMP, 2, CGMP, 3, receptor kinase, okay, 4, calcium. All right, so in the examination, I will definitely ask that question. If you look at the diversity of the endocrine system, hormones are synthesized in a variety of cellular arrangement. And here, down here, in male and female, we have a different types of hormone being released. And of course, you know, they are being released for a specific purpose. For example, like the thyroid gland, it functions to produce T3 and T4. T3 is 3-iodothyronine, and T4 is the thyroxine. Adrenal gland, they functions to produce glucocorticoids and mineralocorticoids, whereas the pituitary gland, of course, when we talk about pituitary gland, you need to know the anterior and also the posterior gland. Okay, anterior pituitary gland or posterior pituitary gland. In the examination, I will ask, what is the hormones being released by the anterior pituitary gland. How about the posterior pituitary gland? Because each of them, okay, either right or left or interior or posterior, they are producing different types of hormones. This picture, you can see there are similarities between male and female counterparts where people have a different types of hormones being released. But the difference is they are producing different amount of sex hormones because they are of different gender. So of course, you know, the ovaries produce mature oocytes and the reproductive hormones estradiol, you know, estrogens and progesterone, whereas in the male, they produce mature spermatozoa and testosterone. And both of them are of different characteristic features, okay? So what this slide is showing is that in some cases, okay, the point number 2, steroid hormones is the precursor molecule for another hormones. In other words, one structure of hormones, they can become like a precursor. Precursor means they are of, or they are the origin, okay, or they are can be the initiator for the formation of another hormones, okay? They are the beginning of another hormones formation. Like, for example, progesterone. It is a female hormone, but it's also responsible to make a new glucocorticoids. It's important for progesterone to be available in the female because for the female to produce the mineralocorticoids, glucocorticoids, and estrogens. Right? In the male, right? The testosterone is important in the formation of another hormone. We call it estradiol and also dihydrotestosterone, right? So this is important for you to know. Even though this is only one structure, it's important for this single structure to be available in your circulation for them to make a new form of another hormone. In this picture, I'm showing like cholesterol. That's the structure of cholesterol. You can see it forms the progesterone. So you know that cholesterol is a type of lipid. That's why we classify this as a group 1, steroid-based or lipid-based hormones. Once you have the cholesterol inside your body, the liver will start to metabolize this cholesterol and they modify some of the chains, making a progesterone. And from progesterone, when they are being synthesized, this progesterone will further form aldosterone, testosterone, cortisol. Not only that, okay, once the testosterone being produced, it also can act as the precursor for another formation of hormones, which is the estradiol. So need to know that the hormones can act independently, okay, based on its own structure. At the same time, it can also become a facilitator, or it can become an initiator or prerequisite for the formation of another hormones, okay? This is another example I mentioned previously. So same thing, you have A, the cholesterol derivative. You look at the structure like 70 beta estradiol, okay, in A under cholesterol derivative, you see the structure is quite similar to the testosterone, right? And also similar to the cortisol and progesterone. In other words, the backbone of the structure or the formula, the chemical structure is almost similar. It's just that where is the addition of oxygen or hydroxyl group or another ring, okay? So this is, you know, the differences, but all of them are originated from the cholesterol. Okay, that's why we call these hormones are cholesterol derivatives. It derived from the cholesterol. Look at B, tyrosine derivative. You look the structure of tyrosine on the top left. You see underneath it says T3. It means that is the 3-iodotyrosine. All right, 3-iodotyrosine is being produced by your thyroid gland. Now I become a bit weird here in the picture. I don't know what happened to my face. Now it's gone. All right, so T3 and T4, almost similar structure, you can see there. All right, and also norepinephrine and epinephrine. They are all derived from the mother structure. We call it tyrosine derivative. We call it tyrosine. For the peptides, it depends on the amino acids or the presence of peptide inside the structure. Some peptide can bind, some amino acid, they can, just bind with another amino acid, forming like 2 polypeptides. Or if, okay, for certain condition, they can also choose to produce a longer chain of polypeptide. The final one, the D1, is the glycoproteins. You can see it's include the TSH, FSH, and LH. So these are the diversity, okay, the different types or different origins of the hormones. I take an example here. You started with the phenylalanine because this is all underneath going to form the norepinephrine and epinephrine, which is your rushing hormones, or what we call the panic hormones. Phenylalanine is a type of amino acid. It being converted into tyrosine. Okay, so from tyrosine, with a certain modification in terms of the structure, an addition of carboxyl group or maybe amino group to its main backbone. So it changed the stability, and also the it renders the activity of this what do we call it, proteins or peptide hormones. From phenylalanine, it turns into being converted into L-tyrosine by phenylalanine hydroxylase. After that, tyrosine hydroxylase convert them into DOPA, L-DOPA, and then dopamine, norepinephrine, and epinephrine. If you look on the left-hand side, which is the chemical structure, the ring, okay, is conserved in each up to the bottom one. Okay, the ring is there. Means that they are from a single mother structure. So we call it as tyrosine derivative. You can also call them as like phenylalanine derivatives. But the most accurate one is the tyrosine derivative, because phenylalanine can also produce or being converted into another form of hormones. Okay, give me 1 minute. Let me try to check if someone's stuck in there. Right, how the hormones being transported in our system. You just look at the right hand side, the image in the, where is it? Here, can you see here, here, yes? All right, it's water soluble hormones, receptor in plasma. You see the secretory cells, it refers to the glands or endocrine glands that produces the hormones. It produces the water soluble hormones, and after that you can see it's penetrating the blood vessel, and from the blood vessel it leaves the bloodstream and enter the extracellular environment, and after that the water soluble hormone binds to the receptor protein, which is on the target cells. So once this combination, okay, the hormones and also the receptor protein bind together, they will become activated. The cells will become triggered to produce the response. The signals that they receive from the binding of water-soluble hormones and receptor protein can be directly initiating the cytoplasmic responses, or they can enter the nucleus and change the DNA and produce a new protein. Okay, the nucleus is in purple. So there are two ways. Once the water-soluble hormone bound to the receptor protein, they can cause responses in these two pathways. Okay. On the other hand, you see on the right-hand side picture, you have the secretory cells. They produce they excrete the lipid-soluble hormones into the blood vessel, but with the transport protein. Still, they need to have this transport protein. Why? Because the lipid-based hormones not only very heavy because they have longer chain, but they are also not soluble inside the blood vessel, because you can imagine the blood vessel now is full of water. Lipid soluble hormones, they don't like water, but they don't have choice. They need to get into the blood system for them to give the effect, right? So from the secretory cells, they penetrate into the bloodstream, and after that, they will go into the cells, and you can see that there's no receptor. Of course, they have the receptor. I think this is on the, no, they don't have. They only have receptor protein inside the nucleus, and they can also change the gene expression inside the nucleus. Inside the nucleus, where the transcription and also translation occurring. What is transcription? The conversion of DNA into RNA. All right, this is happening into the nucleus, and from mRNA or RNA, they will be converted into the protein by a process we call it as translation. Okay, DNA to RNA transcription, RNA to protein translation. All right, so up to translation process, you have a new protein molecules that can act on whatever surface they want, okay, whatever effect they want to have. If you look at down there in the box, feature, concentration, binding, affinity, specificity, this is all just a small information for you to know that the receptors are very low in terms of the concentration, but the transport protein is very high. It's billions per microliter. The binding affinity is very high. I mean, this is for the receptor. But for the transport protein, the binding affinity is very low. It means the binding affinity means how strong they can bind, right? How strong they are being glued or attached to the hormones. Okay, for the receptor and also water-soluble hormones, they are very high binding affinity, means they stick together very strong. But for the transport protein, they're very low, easily detachable. Okay, so that's the characteristic of the lipid and water-soluble hormone. When we call water-soluble, it refers to the peptide. Okay, peptide hormone or protein-based hormones. This is another example of the transportation. Give you example like the bottom here. In the blue one, okay, you see the total hormone T4 and T3. Both of these hormones being produced by the thyroid gland. T4, okay, you can see the total hormone 8 microgram per deciliter, very high. Percentage of total free hormones 0.03%. In terms of the nanogram per deciliter 2.24, molarity 3, and the half-life that they can stay in the blood, it's 6.5 days. But T3 have a very short term lifespan. Okay, they cannot stay in the circulation for a longer period. So this slide is only to show you that the thyroxine, which is T4, and 3-iodothyronine T3, they have a different characteristic. Okay, based on the structure, it renders the activity or the biological activities of these two hormones is changing. This is another explanation, okay? For example, glucocorticoids are transported by corticosteroid binding globulin. You can find the hydrocortisone or simply cortisol also circulates in plasma in protein bound and free form. The main plasma binding protein is an alpha globulin. We call it transcortin or corticosteroid binding globulin, CBG. This is produced in the liver and its synthesis is increased by estrogen. Okay, so CBG binds most of the hormones when plasma cortisol levels are within the normal range. Much smaller amount of cortisols are bound to albumin. All right, so what they're trying to say here is that if you look at this structure on in the bottom right, in the box or in the table, you can see the SHBG and also the CBG. They have different amount and different way in the system. If you look at the SHBG, okay, like the previous one, maybe I skipped the SHBG. It's not here, but maybe next slide. Okay, SHBG is the sex hormone binding globulin. Right? And CBG is the corticosteroid binding globulin. You can see the derivative. Okay, not derivative, the type of hormone that they carry or that they transport. You can see SHBG is the glycoprotein. Okay, they are binding globulin, means they are proteins or transport protein. They can bind or bring, carry, whatever, transport, attached to it only one molecule of dihydrotestosterone. SHBG can carry only 2 testosterone, 5 estradiol, or more than 10 estrogen, more than 100 progesterone, cortisol, and corticosterone. But if you look at the CBG, okay, this is the transport protein. They can carry more of these hormones. So this is very important for you to know that the transport protein have different capability to carry with them, okay, how many hormones they want. So that is also related with the affinity, how strong they can bind and how strong they can carry a certain hormones. Give you example here, like number 1, the oxycorticosterone and progesterone interact with the CBG with sufficient affinity to compete for cortisol binding. Aldosterone, okay, being produced in the form of mineralocorticoid, or known as the most potent natural mineralocorticoids. They don't have a specific plasma transport protein. Gonadal steroid binds very weakly to the CBG because they are bind very strong to the SHBG. So this is another way, okay, for the lipid-based hormones to travel inside your bloodstream. They require assistance. These assistants are the transport protein in the form of SHBG or CBG. And between SHBG and CBG, they have different affinity. Okay, they have different capacity, how much hormones that they can, carry with them. So this is a way that the hormones can be transported to one location to another in order to reach the target cell. If not through the target protein, there is no way that the lipid hormones can travel. Okay, so they will become degraded at that site only. So of course, the transport protein play vital role in transporting or taking up these hormones so that this hormone can reach the target organs. Okay. All right. So this is the hormone action and signal transduction. Of course, the second messenger I've mentioned previously that is occurring inside the cell. But of course, the first messenger is your external stimuli. How do you get the stimulus or what we call initiation, innovation? What do we call? What else? Stimulus or stimulation. It's come from the external sources. If you are exposed to the light, or you are starving during Ramadan, or temperature, heat, or maybe the pH. So these are affecting your signals that you receive or that you produce. Okay, so we always start the hormonal action through these steps. Okay, these multiple steps. We start with the first messenger, second messenger, and after that it goes into the nucleus where you can find the DNA transcribed to the RNA, RNA translated into protein, and protein can give the output. Okay, decide what the cells or what the body is going to do. Okay, so this is the ways or the multiple steps that we have for the signal transduction. For the stimulus 1 and 2, I think this is similar like what have been mentioned previously. But if you look at the recognition, okay, that's the stimulus, the external stimulus, where the first messengers are being released or occurring. After that, you can classify them based on these two groups of hormones, group 1 and group 2, where the hormones is being released. You can see group 1, you have the hormone receptor complex. Whereas group 2 hormones, there are many different signals being produced. It includes the manipulations of the DNA. They're changing the DNA information. Okay, they're changing the transcription process. For example, instead of like if you are what do we call it? If you are hiking or climbing the Everest Mount, okay, as you go up to the tip or to the top of the Mount Everest, okay, you will receive less oxygen and you feel very cold, okay, because there's a higher humidity and also the temperature is reduced. But your body have a special ways how they can create or how they can use hormones in order to increase production of protein that makes your body become heat or become higher temperature. Okay, So it antagonizes. This is what we call, like last time, this is the feedback loop or feedback mechanism. If your body is very cold, or shivering, or like you are trembling because you cannot stand the coolness, so your body responds to it by producing a protein, okay, that can maintain your body temperature, right? So this is different way. And in order to produce that protein that change the temperature of your body, this must go through, or the manipulation, the changes need to happen inside the nucleus. Okay, start with the process DNA, RNA, and then to the protein, whatever protein that we need. This is the single signal transduction. Okay, so this is the example, very detailed example. You start with the recognition. In the case of hyperglycemic, I put it here, your body responds to it by producing insulin, so that the blood glucose level from high, they go down, okay, they will decrease. And in order for that to happen, you know, the insulin molecule is very big, they cannot penetrate the cell. So they need to attach, okay? You see the insulin in oval shape, and then the smaller one like a 11, number 11 there. There's also insulin bind to the special receptor on the cell surface. Once this insulin bind to the cell receptor, they will initiate multiple signals. Okay, so this signal generation can then, okay, send the information into the nucleus, so that the nucleus can change the DNA information and then produce a protein that they want. In this case, you can see the effect when the insulin bind to the receptor. At the bottommost, you can see, all right, the pathway, if it's true, like very sophisticated one, GRB2 MSOS plus P21 RAS, RAF1 MAP kinase, they will initiate the cell growth. Okay, the cell will come, you know, develop, can proliferate, okay, undergo cell cycle. They can also produce a new DNA synthesis, okay, or early response genes. On the other hand, if it's undergo mTOR, okay, P13 kinase or PTEN, so they obviously can initiate a different types of effects, right? If the protein that you are producing is for protein translocation, okay, the protein that is being released is insulin receptor, IGF-2 receptor, and glucose transporter. These are the proteins that is responsible to translocate the protein that have been synthesized. It can also initiate or catalyze the enzymatic activity. It can also change the transcription process. In other words, there are multiple effects once the hormones like insulin bind to the cell surface receptor. Okay, there's not only single effect. Of course, we know that some hormones, they can give single effect, but most of them have multiple capability to produce a different or more effects. So this is another example for you to illustrate this. I don't want to discuss this further, but for you to know that once the hormones bind to the cell receptor, it's initiate a new information or signals in the form of, you know, protein production. And this protein can then affect and decide what they want to do in order to sustain the body. This is also another process I mentioned earlier. From the gene, which is your DNA, you undergo the transcription process, forming the primary transcript. And primary transcript being degraded into mRNA because they undergo some modification and processing. The DNA is the double-stranded. Okay, RNA is the single-stranded. Okay, ribonucleic acid, DNA deoxyribonucleic acid. So simple as that. Okay, once the RNA being produced, it needs to be transported out from the nucleus into the cytoplasm. and into the cytoplasm, the protein will be producers. We call this translation process. The protein will be produced in the ribosomes that is located inside your cytoplasm of the cells. Once the protein been produced, it can, disturb, or it can enhance, or it can improve the activity of the current hormones that you are having. 3 types of hormones. This is the hormones important for carbohydrate metabolism. If we mention about the carbohydrate metabolism, it is important for production of a new steroid-based hormones. For example, here, the tyrosine and also epinephrine. Okay, so this is the example. For hormones that control carbohydrate metabolism, I don't want you to go a bit deeper on this, because this is like the master's or PhD level. Not for you right now, but you can have a general idea and try to understand what you can understand from this one. But it's not going to be asked really in the examination. If I ask, then you will get a bonus mark for this. Okay, remind me on that. Okay, this is just the illustration. I don't want you to go and understand that too much, but just enough for you to know that hormones control carbohydrate metabolism. You need to know that I give the example, 3 example. Okay, like here, epinephrine, glucagon, and insulin. Okay, this is the epinephrine action. It takes time for me to illustrate, but I already put the description here. Glucagon and also the insulin. This is how the hormones can control the carbohydrate metabolism based on these 3 hormones production: epinephrine, insulin, and glucagon. Okay, this is insulin too. Let me discuss a bit on this, okay, so that you're not left behind. So I will take a simple one, like the insulin. I already discussed this, but this is an illustration to show you. On the top one, extracellular space, is where you can find that the insulin bound to the receptor site, like the love shape, like the heart shape. That is the insulin. It binds to the alpha receptor that is available on the outer surface, but the bottom part of the receptor, you have the B or the beta receptors that is attached to the alpha receptors. Once the insulin bound to the receptor site, lots of tyrosine, tyrosine that is available in the tyrosine kinase domain. Okay, tyrosine kinase domains is another portion of the beta receptors that homing or that locating the tyrosine molecules in there. Okay, you know that the tyrosine is the hormones from tyrosine derivatives include what? Okay, already mentioned previously. So once this tyrosine is available on the outer side, or on the inner side of the cells, we call it cytosol. It can facilitate the production of a new hormone, new protein. You look at the point number 3. Once the tyrosine on the receptor are phosphorylated, with the letter P, phosphate group need to be added to the tyrosine in order to activate them. Right? Once the tyrosine on the receptor are phosphorylated or been activated, the receptor then phosphorylates tyrosine on target protein. Okay, so now the protein on the target protein, we call it insulin receptor substrates. which then act as the second messenger to produce a wide variety of cellular effects. All right, you can see here the target protein is empty, right? It's empty now, but then the tyrosine on top of the target protein will bind or will be phosphorylates by the phosphate group. Phosphorylates mean addition of phosphate group at the bottom one. The phosphate group binds to the tyrosine. Only then the tyrosine can bind to the target protein and activate, okay, become activated for whatever signals or whatever activity that they have been instructed to do. Okay, so how does the insulin work? Lots of things with regard to the insulin. Maybe I need to ask you in the essay question for this insulin, okay? Because there's lots of things related to insulin, and in fact, I think insulin is the most appropriate example for me to show you. If you look at the bottom, just imagine like the two cells there. You have the insulin receptor and then insulin molecule, which is the hormones. And then you have the glucose channel, which is closed. But once the yellow one, okay, the insulin bound to the receptor or bind to the insulin receptor, they will open another channel. Okay, the glucose channel will be open to allow the glucose to enter the cells. All right, so this is the prerequisite. Without the insulin bind to the insulin receptor, there is no way the glucose can enter the cells. Right? So of course, it's important for you to have the insulin that is functional, so that whenever you have lunch, these insulin molecules can bind to the insulin receptor on your cells and then uptake or allow the glucose to get into the cells and convert these glucose into the energy or ATP. That is the simple description regarding the insulin. And you can read more here. I already put it here. This is also important for you to understand the effect of insulin in the metabolism. Insulin acts not only in the muscle cells, can act in the liver and also adipocytes. What are the metabolic processes that is involved in this insulin? Okay, the most popular example is the glucose uptake. You're using your muscle every day to blink your eye, to walk, to speak, to eat, to see, to bend, whatever, okay? So you need the muscle. So the muscle have these two proteins, myosin and actin, that are not counteracting, but they are functioning to relax and also to contract so that you can move. Okay, you can relax and contract. In order for the relaxation and the contraction to happen, you need to have this insulin to promote glucose uptake. More uptake of glucose into the muscle cell means that you are producing more ATP, and more ATP means you get more energy to contract or to relax the muscle. This is the example for the glucose breakdown that is occurring in the liver. The effect is increasing. Increasing what? Okay, increasing the insulin, increasing the glucokinase. Right? Target is for, like the top one, GLUT4 transporter is increased because of the insulin bound to the insulin receptor. All right, and some also other example like glycolysis. You can find that the insulin bound to the insulin receptor on the muscle cells and the liver cells. The increases here, it refers go back to the metabolic process, okay, not to the target. All right, correct me if I'm wrong. The effect here means increases what? Increases glucose uptake. The effect increases glucose breakdown. What is the effect that increase? The glycolysis. What is the one that decreases? Glycogen breakdown. So the glycogen breakdown effect is decreasing when insulin bound to the insulin receptors. Okay, this is example of the metabolism related with the type 1 and type 2 diabetes. If you look at the top one, glycogen and we have this pancreas. Decrease insulin in the blood vessel due to the genetic factor. We call it congenital type of diabetes. Okay, occurring quite early in the child, where you can find the insulin is decreasing. And after that, the effect of low insulin means they cannot move quicker. They have like a muscle weakness because of this low insulin. All right, they don't have enough insulin to unlock the glucose channel. So what happens is that they cannot survive longer, okay? They have some complication and they require a treatment, means that they need to be injected with a constant insulin injection. Whereas for the type 2, you can see that the pancreas is sufficient insulin being secreted. Yes, they have enough like you, but some of us, okay, some of us, the insulin that is being produced, they are not functional. They are unable to bind to the receptor. Okay, they are not able to be bound onto the insulin receptor. What happens is that again, muscle unable to use the glucose because the insulin is not functional. In other words, we call it insulin resistance. What makes this thing happen? Your lifestyle, like obesity, or some, your eating pattern, right? So these are rendering the activity of the insulin. The insulin become less active if you have obesity and if you have some other complications. At the same time, people with type 2 diabetes also have an increased risk of Alzheimer's. That, people theorize that the increasing number of people getting Alzheimer's disease could be tied to our fast food, high sugar, diet, lifestyle. So this is all the binge, not so good lifestyle. So we are not promoted. We are not, promoting this in our life. Okay, you need to take care of your blood glucose level. So this is the association between the insulin and sport. You can just read this one because for the knowledge, you can see the trend. Okay, the trend while in sport, while you have like activity, you can see the insulin and also the glucose strengths are varies, right? Because you are consuming lots of energy. Consuming lots of energy means you need more ATP. More ATP means that you need to take more sugar, okay, or glucose. Without the glucose, okay, you cannot perform this sport or activities. But how is the method for you to allow glucose to enter your cells? Of course, it's through the insulin. So the insulin secretion depends on what type of activity, how strenuous the exercise, okay, how hard the exercise are. If it's like very, not so intense, okay, just like for the fitness, maybe you're jogging or skateboarding, cycling, the glucose strength like, going down, all right, because you're not consuming too much. But if you are like football, volleyball, okay, this kind of like giving mix, because sometimes you need to score, that's your turn. Sometimes you pass the ball to your friend and you kind of relax a bit. So this is like a mixture of the insulin on and off insulin secretion. For the anaerobic, where you need a high spike of insulin, only that particular point, like the heavy lifting, or maybe like, I think, don't know what is this? This is like a kung fu, something like, okay, where you need a high sharp spike of insulin secretion. Okay, so really depends on the demand, what type of activity you are doing, and that can change the pattern of the hormones or the activity of the hormones. All right. Okay, so this is the hormones differences between between enzymes and hormones. You can have a read through, because I also gonna ask you for this one. Okay, this is also important. The difference between the hormones and enzyme. What are they? Okay, hormones, basically they belong to the endocrine system, but enzymes, okay, some of them being produced by the glands too. Okay, but the glands is duct glands, means they have a special channel. They travel inside the blood. But the hormones, they can choose whether they can, you know, act on within the bloodstream or outside the bloodstream. Hormones, they travel through the blood. Enzyme, they can also travel through the ducts. Okay, and some of them can travel through the bloods too. Okay, but most of them through the ducts. Hormones are very low in quantity. Enzymes very high. Reaction of hormones, okay, because they are very less in quantity, maybe the reaction is very slow and the reaction is fast in enzyme. Hormones involve metabolic activities, means the breaking down and also of certain food products, or synthesis of a new proteins. Enzymes involved in digestion, like lipase, amylase, pepsin, okay, hydrolase. So these are all the enzyme. Enzyme means they want to catalyze the reaction. They want to have converting a certain compound to another, you know, new compound. But the hormones is, you know, even though it's slow, but it's important to maintain a good metabolism in your body. Okay, when we mention about metabolism, always remember, just like you know, example like the diabetic one that I've mentioned to you earlier. Okay, so this is the summaries. I hope you can read through this one too. This kind of summarize everything that I have showed to you earlier. I think that's it for tonight. Thank you very much. I think I've spent around 1 hour with you. Sorry for a bit late for today's session. Okay, so if you want to have the recording for the previous lecture, you can always go to the YouTube and go to my channel. I will upload the first session, like the hormone one, and for this one, I will try to update after this. Okay, so you go to the YouTube, type Tengku Faris, and go to my channel, and don't forget to subscribe. No, I was joking. Okay, no need to subscribe if you don't want to, because I'm not collecting any money from it. It's just only for educational purposes. You can have a look if you don't understand anything. If I'm just, you know, making my words, please, if you want to ask me any questions, if you don't understand, we can always have another session for me to clarify, because the slide is quite lengthy and I don't want to spend most of your time for tonight. Thank you very much, everyone. Hope to see you again. If you have any question, feel free to ask me now or in the next meeting or next lecture. All right? Is there any question?
Doctor,
Yes.
I want to ask for the attendance. We cannot click in for attendance.
What happened to your attendance? Maybe it's your computer.
No, everyone cannot turn in. There's no